Thanks, Susan. As Susan mentioned, I'm Annie Murphy, and I'm a PhD student at VIMS. And I'm going to try to give you, can everyone hear me okay? I'm going to try to give you a brief snapshot of my research at VIMS, specifically looking at how commercial clam aquaculture can remove unwanted nutrients from Chesapeake Bay. So just a brief overview of nutrients in the bay. We know that Chesapeake Bay, the watershed of Chesapeake Bay is relatively large, and activities on land such as wastewater treatment plants, urbanization, large-scale agriculture, those types of things are all contributing a significant amount of nutrients to the water and to the bay. And too many nutrients can cause eutrophication to occur. And eutrophication is the increased supply of organic matter. And it's associated with a whole suite of negative impacts in the water, including harmful algal blooms, low oxygen zones, and overall a poor water quality. So for the past decade or more, we've been trying to figure out ways to reduce the nutrient loads into the bay. So EPA has implemented the total maximum daily loads, or TMDLs, in efforts to reduce nutrients. And local governments and industry are trying to figure out best management practices to reduce these nutrients. So how do shellfish fit in with nutrient cycling? When I say shellfish, I typically am talking about bivalves, such as oysters, clams, scallops, and mussels. And all of these bivalves are filter feeders. And I like to think of filter feeders as nutrient transformers. So they're transforming the nutrients that are found in their food, the phytoplankton, into different pools. So the shellfish will feed the phytoplankton and clear the water of the phytoplankton, and they use the nutrients in the cells of the phytoplankton to grow, so they get bigger. They also produce waste, and so in the form of biodeposits, which are another form of particulate nutrients, including nitrogen and phosphorus. And these shellfish are also directly excreting waste in the form of urea and dissolved nitrogen and phosphorus, ammonium and phosphate. Additionally, these biodeposits are really juicy, and they're great for microbial communities to degrade and decompose these particulates, and so there's more dissolved nutrients coming from those microbial processes. So all of this dissolved nutrients going into the water column is fueling additional plant production. So it can fuel more phytoplankton, which is great for the shellfish because that's their food, sort of a positive feedback, but also can feed seaweed or macroalgae, which is often associated with aquaculture operations, so the nets and the cages that they use in the industry. So it can get pretty complicated relatively quickly. So we have to think back of our main question. Can bivalve aquaculture effectively reduce nutrients? So there's three main ways that I see bivalve aquaculture reducing nutrients. The first is relatively easy, so harvesting. Shellfish are grown by planting juveniles or small organisms directly into the environment. They feed on the phytoplankton. They assimilate or incorporate that nutrients into their tissue, and then you remove the adults, the larger clams or oysters, from the system. So in the tissue, there's nitrogen and phosphorus, and you're removing that. The second way is denitrification. Denitrification is a microbial process that essentially it converts available nitrogen, available to phytoplankton and macroalgae growth, and it converts that into N2 gas, which is not readily available to plant growth. And so the biodeposits may enhance denitrification. The conditions have to be just right for this to happen, and this is clearly way more complicated than just this simple arrow. There's a lot of steps that are involved. The third and final way is through macroalgal harvest. So as I mentioned, macroalgae are often associated with clam aquaculture operations. And just like this first way of harvesting the actual target product, macroalgae have also incorporated this nutrients in their tissue. So when we think about these three kind of pathways of removing nutrients associated with aquaculture, we also have to remember that, as I said before, the bivalves are excreting and the biodeposits are being decomposed. So there is a risk of local eutrophication, nutrients coming directly from these operations. So my research in particular is on the hard clam industry in Virginia, and specifically on the Eastern Shore. It's a very lucrative industry, and we actually lead the U.S. in clam production. And basically the growers go out and they plant juvenile clams directly into the sand, into the sediment, and they have to use this plastic mesh net that they place right on top of the clams and on top of the sediment, and this is to protect their product from being eaten by things like blue crabs and rays specifically. So this net is actually a perfect place for seaweed to grow. And they recruit to the nets, and they produce more biomass. And we went out and measured the amount of macroalgae on the nets seasonally. So this is what this graph is showing. And as you can see, in July, it has the highest biomass. So in the summer, the macroalgae really are blooming, and this macroalgae is detrimental to the clams. So it can be sort of considered a harmful algal bloom if you think about it. So too much macroalgae on the nets will start to smother the clams. It's not good for growth. The water flow is severely reduced, and water flow is necessary for the clams to feed, delivering phytoplankton and oxygen to those clams. So the growers go out and they have to sweep their nets periodically. And they use this big hydraulic brush, and they kind of lawnmower over the nets and sweep the macroalgae off. So we were interested in how clam aquaculture impacts nutrients and what the role this macroalgae is playing. So we went out and we did an experiment where we had 3 treatments. One was clams with macroalgae. The second was clams without macroalgae, and finally a control bear site. And we conducted this experiment both in the light and in the dark. And we went out in May, July, and October to determine seasonal variability. So basically we went to Cherrystone Inlet, which is on the bay side of the Eastern Shore. We have these core tubes, which are acrylic plastic clear tubes that we kind of just shove into the sediment, cap them, and we sample for nutrients, oxygen, and carbon dioxide over time, and we can figure out how the benthos, or the sediment and the clams, are affecting the water column. And so we found that this graph just shows fluxes of ammonium from the sediment into the water, and we found that the clams, which are in the red bars, are significantly higher than the bare sediment, which you can barely see the flux during each month. When you add macroalgae, this flux decreases. So what's happening is the macroalgae is serving as a cap, and it's taking up all that nutrients that's coming from the clams and coming from the sediments, and assimilating it, incorporating it into the macroalgal biomass. Similarly, we found for phosphate, or dissolved phosphorus, we found that the clam beds again were a significant source, and macroalgae actually don't have a significant impact on this flux. So when we added the macroalgae, it did not decrease the flux as much. So the macroalgae are taking up the nutrients that are fluxing from the sediments. It's important to note that this storage of nitrogen and phosphorus in the tissue of the macroalgae is not permanent. Eventually, the macroalgae will get so dense that it will die off. Self-shading can occur where it can no longer get sunlight, and so it starts dying off. Increased temperature will also cause die-offs. And so this dead organic matter is great for microbial degradation, and the microbes start going to town on this material, and they use up oxygen, they release the nutrients that was sequestered in that tissue, and now we have a risk of local eutrophication. And so it's necessary to actually remove the macroalgae from the system to consider it an actual permanent sink. And so a polyculture that integrates both mackerel algal harvest as well as the clam culture may be feasible and ecologically beneficial. The problem is that there's no macroalgal market, especially in Virginia. So these are just a couple of potential markets for the algae. The nutrient trading credit programs may play a role in this, so providing the growers an incentive to actually remove the algae. Also organic fertilizer, so in other regions of the country, including Hawaii and other states, they actually use macroalgae as organic fertilizer. And finally, seaweed, the new vegetable. So the movement and the push to eat sustainably and buy locally is kind of pushing people to start thinking about consuming seaweed in our country. And so basically, in conclusion, clam aquaculture, the clams are converting phytoplankton into biomass, growth, and they're also converting into dissolved nutrients. And this nutrients may lead to local eutrophication, but it's being sequestered temporarily by the macroalgae. And so by removing the macroalgae, we can further reduce the risk of local eutrophication. Clearly, there needs to be more research and more research into the economic feasibility of harvesting the macroalgae and developing incentives for the growers to do this. So I would love to take any questions. They're about 4 hours. So the oxygen drops quite quickly when you're isolating this small little ecosystem, and the clams are breathing, respiring. So we let it go for about 4 hours. Yeah.
Have you considered seeding the mesh with algal spores that would give you economically valuable algae like chondrus or other things that produce polysaccharides that can be harvested, for which there is a market?
I have not considered that, and I don't think the growers are considering that. It's really just, in their mind, it's just a nuisance, and so that would be a good approach to kind of get more of a monoculture of some species that you actually want to harvest.
Estimate of the volume of algae produced?
I think on a single farm, it depends on the season. Something like, I don't know, 13 to 20 pounds at any given time. I've done some calculations based on uptake rates, but yeah, it depends on how frequently they sweep it as well. So you sweep it, and the uptake rate is density dependent. So the growth they'll grow really quickly, and then they'll start to senesce. And so, yeah, there's a lot of variables, but definitely something that I'm passionate about and interested in looking more into. So,
all right, one last question.
This is going to sound silly, but can you feed it to chickens?
Chickens kind of complete the loop, right?
That's right.
Yeah. I'm not an expert on chicken nutrition, but that would seem reasonable.
Thank you, Annie.